Hi, everyone. My name is Alejandra Cervera, and first I would like to thank the organizing committee for allowing me to present my work here today. I'm going to talk about an RNA-Seq pan-cancer panel approach that we have been developing at INMEGEN with the Cancer Diagnostics Lab. And although we have a B-cell acute lymphoblastic leukemia presentation before, I would want to give a short introduction. So acute lymphoblastic leukemias is the most common type of childhood cancer, and the B-cell type is the leading cause of cancer-related deaths in children and young adults. Sadly, the incidence in Mexico is quite high, and also the survival is low. So the incidence is also high in Latin Americans. This has been also observed in Hispanics in the US. And unfortunately, the 5-year survival currently in Mexico is around 60%. The reasons for this are many. Some are related to lack or misdiagnosis, some to abandonment of treatment, death from toxicity from chemotherapy. And also we have observed that Mexicans or Latin Americans in general present with a high risk molecular subtype at diagnosis in higher proportions than other populations. These molecular subtypes that are associated with the risk, the World Health Organization recognizes 13 of them, and I have grouped them here based on the genetic alterations. So the first group is based on specific mutations. Specific fusion genes, I'm sorry. The first one is the BCR-ABL1, which is a very famous fusion gene known as the Philadelphia chromosome. And then we have other specific mutations. On the second group, we have phenotypes that look like these specific mutations, but they have a different array of genetic alterations. So lots of fusion genes, also specific mutations like we saw in the previous talk. And then we have another group that's related to chromosomal and gain losses. And finally, we have two groups for the patients that cannot be diagnosed in any of the previous ways. And usually this diagnosis is achieved either to FISH, RT-PCR, or karyotyping in the clinic, but that only works for the blue groups. And for the green subtypes, we have to do NGS because of the large amount of genes involved in any of these subtypes. And of course, you cannot say that you didn't find anything if you don't do NGS. So what we're trying to do in this project is come up with a solution that would be a single assay to diagnose these children and give them their subtype. This is important because the therapy is related to the subtype, and there's no way these children will receive targeted treatment if we don't characterize them. Our approach includes an RNA-Seq panel of 1,300 genes. The idea of doing a panel of genes is because we think this is going to be more likely to be adopted in the clinic in Mexico. It's expensive to do sequencing, so we don't expect them to do whole transcriptome sequencing. And the idea is to classify as many patients as possible with these subtypes, especially the Philadelphia-like, because it's the most prevalent in Mexico. Disadvantage, of course, is the cost, and that maybe we cannot classify all patients in this manner. We already started this project. It has 113 patients already sequenced with this panel of genes. We have been able to identify 25 different fusion genes. This gave us a classification for 40 patients, so out of these 113, and these patients already have their classification. And here we can see a summary of these fusions based on the different subtypes, and especially based on the prognosis. So here we see what I was telling during the introduction, that we have a lower proportion of good prognosis patients. The green bars are what we expect based on other populations, and the blue bars are what we observe in our own cohort. And here we have the Philadelphia-like, which is poor prognosis at diagnosis, and we have a higher proportion of these patients than expected. And unfortunately, this blue bar is expected to grow even further because this classification is only based on the fusion genes, but we know there's other genetic alterations that are associated with this phenotype. One of these specific alterations is overexpression of CRLF2. And this in the clinic is usually diagnosed with RT-PCR. And if you have whole transcriptome, then you can do TSNE or something else. And what we did here was just use the K-means algorithm to find the groups of expression. And here we see that this C4 group is the highest expression in the patients. And we were happy to see that we can actually identify all the genetic abnormalities that push the patients to have this level of high expression of CRLF2. So here we observe this fusion gene, this fusion gene that we can see that we have a patient that is not in the highest group. This is consistent with literature. It has been reported that this fusion drives overexpression, but not all the way to the highest level. So what we think here is that the mutation is the one that is actually pushing the overexpression. And we have here another patient with a mutation, but it has a low variant allele frequency, so it's not driving the overexpression all the way to this level. Here we see all the fusions, and we see that the group that is driving the highest expression is actually duplications inside the CRLF2 gene. This is not easy to detect with all fusion calling algorithms, only with some of them. And then there's this last one that is a fusion that doesn't include the CRLF2 gene, but it has already been associated to this phenotype. And in this cohort, we have observed it once, but now we have some more sequencing data, and we have another patient with that fusion. And well, apart from showing you what we've done of what's already known in literature, we have also been able to obviously explore this data, and we identify that there is a group of patients that have overexpression of MET12, and interestingly enough, none of these patients fall into the known categories of the known subtypes. So we think that maybe this could be a new subtype, so we're working on this hypothesis. So the first thing we did to further explore this is to do differential expression on the patients that have a high expression of MET12 and low expression of MET12, and that they don't belong to any of the known subtypes. And what we see here is that we have a cluster of high expressed genes for the patients that have high MET12. And what's interesting about them is that some of them are located next to MET12. So what we're thinking is that this MET12 overexpression is actually an amplification of a region of chromosome X. In B acute lymphoblastic leukemia, there is also reported amplifications of chromosome X, but in a different region. So this is not reported before. And what we did next is to look into it in a new cohort to see if what we observe in our own cohort is also in some other one. And we use the target cohort. And what we see here, it's a little bit similar to what I just showed. We see that we have, this group here that has a high expression of MET12, and most of these patients here do not belong to these subtypes. Unfortunately, for this cohort, I only have this characterization, but most of them seem to not belong to the three main classifications of B cell. So this could be also an inclination of that. And of course, it would be really interesting if we could correlate this with clinical outcome. And well, so the intervals, I observed that the confidence intervals overlap, but I think it's still interesting and worth looking into because this target cohort is a high-risk cohort. So we don't have here patients that are expected to do well at diagnosis. So I believe that it may be with a cohort that has better survival, maybe we could see a higher difference a larger difference between the patients that have upregulation of MET12 and downregulation, because it seems that MET12 is a good prognosis indication. So we're now looking into checking our own clinical data. We don't have yet done that because these 113 patients, some of them are still ongoing treatment, so we don't have their outcome yet. And we're continuing to work on the other classification, and of course, we would like to explore further this MET12 or chromosome X amplification. For that, I'm looking for students. If anyone is interested in working in this project, you can contact me. This is my contact information. And of course, here I would like to thank my collaborator, Dr. Carmen Alaez Berzon. She invited me to work in this project, which is sponsored by CONACYT and PRONACES. And of course, all the members of her lab, which they were in charge of all the wet lab that was done in this project. So thank you.
You presented in one of your slides this disproportion in the number of cases for children in Mexico. Do you think there is a molecular component, or what is your thought about it?
Yeah, I forgot to mention, but we actually don't know why Mexicans or Latin Americans in general present with a higher proportion of these poor prognosis cases. So of course, one explanation in Mexico would be environmental causes, but that doesn't totally explain this higher proportion that is also observed in the USA in Hispanics. So we think that there's also a genetic component involved in predisposition, but as far as I know, this is not known yet.
I have one question. How much does it cost to perform this panel? I think it's cheaper than sequencing, but in fact, I don't know the cost.
Yeah, it depends. Right now, this panel is being run inside a clinical lab, so it's not like really high throughput. So it's like 8,000 pesos to run this. 8,000, yeah, 8,000 pesos. Yes, 8,000. But if this would be done like more, like the idea of this project is to convince hospitals and clinics to run the panel, and we're working on the software that would be one click and then you get the classification so anyone can run it. Then it would become cheaper because then you will get like all the samples and run them together. So right now which I don't think it's that much considering the improvements in survival that you can get.
Yeah, of course.
Well, something that about the last question, the main doctor, but well, you said that there could be a genetic component that produces this effect in the population of Latin America. But well, that's curious for me because, well, after all, Latin America has a large genetic diversity. I mean, they have an European, indigenous, and African components, many other components. So, in terms of genetic diversity, it's a bit problematic. This hypothesis. I mean, for example, in populations like in East Asia or in the Middle East, where the genetic diversity is lower, well, it could be more easy to propose that a genetic component could be the responsible of this effect. Maybe there could be, as you said before, environmental factors, but genetic could be a bit. I think it could be a bit problematic from that point of view.
Well, it's true that the results are not the same throughout Latin America. So some countries are doing better and some countries have slightly lower incidence. So mostly the survival, I think it's explained through lack of proper treatment for these patients. But still, we do have a lot less of the good prognosis at the beginning. And I mean, I don't know, but I think there must be something genetic as a predisposition, but not, of course, throughout all populations in Latin America. I mean, you have to group them somehow, but I mean, we all know it's a very heterogenic region and we need to look. I mean, in Mexico, even the survival in Mexico City is much higher than in other parts. So Puebla, Tlaxcala, and Oaxaca are being observed as a specific region, but it's also because, I mean, there's been a study that shows that the survival is related to the hospital where you're treated. If the hospital has less than 6 patients per year, then the survival is around 20%. If the hospital has hundreds of patients per year, then the survival is much higher. So it's a very complex problem that needs to be observed from all perspectives.
Have you seen if there could be a new relationship between that and, for example, the lifestyle of that individuals?
I mean, yes, there's not only lifestyle, but there's been correlations with if you live next to a river that's highly polluted, or there's other environmental factors. So of course, poverty is an indication of it's bad for all cancers. So yeah, I mean, environment is a strong component, but then we need to see if we can actually correlate it with the specific fusions that we observe, because one is how the disease progresses and the other one is what you see from the beginning. So we don't know. Yeah, I mean, it could be not genetic predisposition.
Thank you.